Hi there, this is Zihin Zhu and Alicia Kavalin from the University of Michigan MCDB 427 Molecular Biology. In this video, we are going to introduce you to the figure 10.19 in our textbook, Molecular Biology, where French scientist Benoit Chambon characterized the role of TATA box in the context of transcription. This experiment is significant because it demonstrates that the TATA box is important for positioning, where the transcription start sites are for this promoter. Before we dive into our topic, here's a roadmap to our video. We will start with a brief introduction to transcription in eukaryotes, following which we'll be talking about the rationale for the experiment. What did they know back in the 80s, and what were their driving questions? And then we'll be talking about the technical details of the experiment, including how did they generate the promoter deletion constructs and the assays they used. And finally, we'll be analyzing their experimental results together and summarizing their experimental conclusions. So, without further ado, let's start with a brief overview of transcription in eukaryotes. For a eukaryotic cell to function, transcription, the process of making RNA molecules from DNA molecules, has to take place. As you may know, at the beginning of transcription, RNA polymerase, shown here as RNA pol, and other general transcription factors will associate with a specific region of the DNA molecule termed the promoter to initiate the whole process. And then the RNA polymerase will move along the DNA molecule to synthesize the RNA molecule. And now we know that the region the transcription machinery initially binds is called the promoter, and it's a complicated structure consists of different cis-acting elements. And we're just going to focus on TATA box, which is found in many but not all promoters. Back in the 80s, these two good-looking French scientists, Benoit and Chambon, did not know as much. They were aware of the presence of the promoters and the TATA box, but they had no idea what does TATA box do. The questions Benoit and Chambon were trying to answer were twofold. They wanted to know if TATA box determined the transcription start site for transcription, and also if TATA box is essential for transcription. To address these questions, Benoit and Chambon looked at the transcription from SV40 promoters from wild type and several mutant constructs. So on this slide, we'll go through the different deletion constructs they used for the experiments. So the first one you're seeing here is a cartoon of an SV40 promoter. The TATA box is labeled in red, and the three G's with arrows are the transcription initiation sites. The researchers then cloned the promoter region into a plasmid, and thus we have the construct PSV1. For the construct P1A, they got rid of the part downstream of TATA box encircled in the blue box, and then religated the two ends together. Same rationale applies to deletion construct AS, HS0, HS3, and HS4. Then, to answer the second question of whether TATA box is essential for transcription, they got rid of the whole TATA box in the construct HS2. After generating these plasmids, Benoit and Chambon then transfected them into cells. And then they extracted the mRNA and subjected them to S1 mapping to figure out the transcription initiation site. For the technical details of the method of S1 mapping, please feel free to refer to our course video. And here, I'll briefly describe how you get a specific radioactive DNA band for the experiment. So the cartoon is a generic promoter region. I've indicated the transcription starting site with the blue arrow. And then, after the transcription and the mRNA is extracted, a radio-labeled DNA probe, shown here in yellow, will hybridize to the mRNA. With the help of nuclease S1, as you can see here by the blue little Pac-Man, we digest out the unhybridized nucleic acid. And then we can figure out the 5' of the mRNA by looking at how long the radio-labeled DNA probe is. This is important because this can help Benoit and Chambon answer where the transcription started in their construct. And shown here is another construct. Suppose it starts with a more downstream transcription start site. And the same process will happen. You get an mRNA, you get a radio-labeled DNA probe, and then you digest them. And then in this case, the S1 nuclease will thus chew up more of the unhybridized nucleic acid, and you get a shorter DNA probe. Ultimately, we denatured the nucleic acid hybrids, and we ran the gel and performed an autoradiography to visualize the probes. And before we segue into the actual figure, I want to summarize their experimental conclusions first. What I found was transcription from the wild-type SV40 promoter in these two constructs started at the same place regardless of whether the promoter is on SV40 or on a plasmid. And the distance is about 25 base pairs from the TATA box. And before I touched upon the deletion mutant, just so you understand the cartoon, I added extra DNA to the end of the construct, shown here in lighter yellow, so that I can better illustrate my point. In all other deletion constructs, as you can see here, the distance from the TATA box to the beginning of transcription start site is always about 25 base pairs. And thus, one major role of the TATA box seems to be to position the correct start of transcription. Additionally, for the construct that does not have a TATA box at all, transcription will start at multiple different locations. Alright, ready for the real deal? Here's the actual figure. I know it's quite messy, but just wait for it. I'll go through it lane by lane. Let's start with this. The lane marked in red, labeled MA. This is a size marker, and this lane simply serves as a size standard for the whole gel. Alright, and let's move on to the two wild-type constructs. And here are the constructs for your reference. And the results tell us on the gel that the mRNA start at the same location. We can see 2 bands on the gel. However, the researchers indicated 3 on the construct. But as you can see here, the bottom 2 transcription start sites are very close to each other. So chances are they will not resolve well on the gel, and that's why we can only see 2 bands. And before we talk about the different mutants, I want to show that wild-type construct to your left for your reference. And then for the different mutant construct we saw before, the bottom line is the number of base pairs deleted is how much shorter the S1 products are on a gel. For example, for this P1A construct, if it deleted 3 base pairs, on a gel the S1 products were 3 base pairs shorter than a wild type. And the same pattern goes on and on for construct AS, construct HS0, construct HS3, and construct HS4. And this pattern demonstrates that transcription initiation always starts at the same distance from the TATA box. And finally, the second question driving the experiment is whether the TATA box is necessary for transcription at all for this promoter. And remember, here's the construct Benoit and Chambon used for the experiment without a TATA box. And as you can see from the gel on this lane labeled in red, that TATA box is not required for transcription for this SV40 promoter. In fact, transcription started at 8 different starting sites, as you can see here by the 8 bands on the gel. And finally, to summarize the figure, TATA box serves as a yardstick for transcription start site, which always start 25 to 30 pace period downstream of TATA box. And for this specific promoter SV40, TATA box is not essential for transcription. Again, thank you so much for watching, and I hope after watching this video, you will have a better understanding of the figure 10.19 in molecular biology. And go blue!